for coming out tonight for our lecture series talk. Tonight I'm pleased to introduce Dr. Art Laganowsky. He got his BS and MS degrees in biological sciences at Cal State and his PhD at UCLA. He did postdoctoral work at the University of Oxford, and he is a member of the Department of Chemistry here at Texas A&M. Dr. Laganowsky's research is at the interface of chemistry and biology. In particular, he is interested in studying interactions between membrane proteins and lipids. Tonight, Dr. Laganowsky is going to talk to us about biomolecular machines. Please give a warm welcome to Dr. Laganowsky.
All right, well, it wouldn't be official if I don't do a howdy as well, right? So howdy. Okay. So it's a pleasure to be here. I'm going to talk about some of the work that's happening on campus in my laboratory, in addition to teaching most of you or some of you. My goal of this talk is to try and bring in what you're learning in fundamental chemistry into biological chemistry, right? And how we use those fundamental principles to understand how biological molecules work. I'll talk about this in terms of biomolecular machines. These are machines that carry out functions in cells. You're here because of those machines carrying out their processes, all right? So that'll be my goal. I'm going to give you actual research slides that I would normally present, but I'm going to give it to you from the perspective of a first-year student, right? How does the first-year chemistry come into play and drive our research to understand how molecules work, all right? And they didn't give me curtains, so I will start with curtains. Right? All right, so my group's interested in a series of proteins that are embedded in our biological membrane, and I'll come back to these and show you a little more detail. We're interested in these because they're essentially our conduit from the outside of the cell to the inside of the cell, right? And they transport molecules, they're involved in signaling, right? Recognizing, for example, a molecule outside of a cell and telling the cell to grow and proliferate. We're particularly interested in these because they're known as drug targets, right? A lot of drugs are designed to target these and correct dysfunction, right? When proteins go wrong or give disease, okay? We're particularly interested in these things called lipids, right? Some of you may not learn about these till later on, but they're essentially molecules in a cell that form the bilayer, and we're interested in the chemistry of these lipids, right? We're in the Department of Chemistry. All right, if I were to grab one of these lipids with these tweezers and show you in more detail what it looks like, right? Essentially, this lipid you may learn as a ball and two sticks, but you're Aggies, so you're going to know that there's actually atoms there, right? And there's chemistry. I'm not going to go through all of this, but everything you'll learn from here in organic chemistry about different types of structures, whether it's an unsaturated bond, for example, that can adopt a cis or trans configuration, stereochemistry, right? We're particularly interested in how this chemistry of these lipids, right, influence the proteins that they're around, okay? Some of these lipids have barcodes. They actually have patterns which they get modified and tell a cell, right, to either activate or turn off, right? And we want to understand how this pattern of phosphorylation or this barcode is recognized by proteins in the bilayer, all right? So in today's talk, I'll talk about a lot of these different chemistries that I'll give you different names, but all I want you to walk away with is their different structures, right? Different chemical formulas, right? And we as biochemists want to understand how they actually influence the proteins in a cell, right? Today I'll talk about some work that will come back to this famous thing that you've probably seen on your slide. If you're in Hildenfeld's, would be on the wall, right? The periodic table, right? We'll be interested in this first row of transition metals, and in biology, some of these, such as copper and zinc, right? You would not be here without copper or zinc. In fact, all living organisms need copper and zinc. And I'll show you how our work is starting to understand how copper and zinc are coming into play in the bilayer, all right? And then I'll transition into this story about a transporter, okay? I'll start off by talking about proteins. You'll learn about this. It's essentially a polymer. It's essentially amino acids strung together, and these amino acid string or polymer folds up into a protein. And I'm going to show you that in what we call this cartoon representation. And all I want you to walk away at this point is that we have a transporter that's now in this open state, right? And it can change structure. And this state, it can go from this structure all the way to the right, okay? And we're particularly interested how this structure determines how it influences with other molecules, whether that be a lipid or a drug. Maybe we want to find a cure for a disease. And so today I'll talk about how our work is trying to dissect, or better decipher, how binding to this particular state, for example, this lipid, how tight is this interaction? And in chemistry, we refer to that as our equilibrium constant, or better as delta G, right? Gives free energy. And we want to measure this and understand the interaction. In addition, what we want to do is understand how this molecule may interact with the other structure, right? And if these molecules interact differently, right, just like you learned in a chemical reaction, maybe this is my reactant and this is my product. We want to understand how this particular molecule is driving this reaction and influence the equilibrium, right, or the abundance, right, at an equilibrium position at a given temperature, right? So this will be the focus of my talk, focusing on this machine, this transporter, and I'll show you more details of how it works, okay? All right, for those that want more than what I'm going to give you, here's actual references that have been published. There's one more that's under review right now in the final stages of being revised. And you can go to these and read at much more detail than I'm going to go in today. But these are actual scientific papers that have been worked out and published here at A&M, okay? So I'd encourage you to take a look at what your campus is producing. All right, we're interested in this particular problem, right? We're interested in proteins involved in cancer, human disease, and part of the group is interested in proteins that are associated with bacteria. On the left is an electron micrograph of a gram-negative bacteria called E. coli. By the way, everyone in this room has it in their gut, okay? Sorry. But now these rod-shaped cells, or single cells, the longest dimension here is on the order of 1.6 microns, right? Some of you know that you can convert microns to meters, and that would be 1.6 times 10 to the minus 6 meters, okay? Really small, right? And the problem with these bacteria and other gram-negative bacteria is they're on this watch list of potential bacterial pathogens, right? And it's estimated in 2050 that the leading cause of deaths will be due to bacterial infections, and that's because of antibiotic resistance, right? So you and myself need to start thinking about how we're going to develop new drugs, right, to essentially kill these things so that we can live past 2050, all right? And that's the goal. And if we look at one of these cells, we can look a little bit deeper and see what they're composed of, right? And so these are unicellular, just one cell. And inside this cell, here's a watercolor painting from David Goodsill. These bacteria have two membranes, right? We have an inner membrane and an outer membrane, okay? Now on the outside, if you look at these paintbrush-like structures, these are lipids that are decorated with sugars, all right? Why do we care? Well, why should you care? You should care because these molecules called lipopolysaccharide are the ones that help bacteria form this impermeable barrier to drugs, right? Our drugs don't work. They can't get past this bilayer. Now, if we can actually get rid of these molecules, we can now make drugs that penetrate and enter the cell and can kill it, okay? By the way, this molecule of LPS was awarded to Christian Wrights the Nobel Prize for its discovery as endotoxin, all right? So if you get a bacterial infection, your body recognizes this molecule and generates an immune response, okay? So we're really interested in this problem, and we're interested in this bilayer, as I mentioned in the beginning of my talk. And if we were to zoom at this inner membrane, what we would see is lipids. These are our lower or bottom and our top leaflets. This is what forms our bilayer, right? Essentially, think of a balloon. This is the membrane of the balloon that keeps it inflated, all right? And now we have membrane proteins that are embedded in this bilayer, right? And their role is to either bring molecules from the outside in or outside in the other direction. Now, importantly, you're seeing me right now because of a protein embedded in the bilayer that's actually being activated by light, undergoing a cis-trans isomerization that's now turning on a signaling network. You're hearing me right now, if you thought about this, by a protein in the bilayer that's sensing the vibration in your eardrums, right? Biology and biochemistry is all around you. All right, so we're interested in these proteins that carry these really important roles, and more importantly, a lot of drugs target these proteins, okay? If you go to the store right now and walk down the aisle, look at in the pharmacy or the drugstore, a lot of those molecules that are in those packages are targeting these proteins, okay? Super important. All right, I'm going to lead into now this transporter, and I'm going to give you an overview. I don't want to give great detail, why we're interested in it, and what does it do, and how does it work, okay? So we're interested in this transporter, and its role is to take a lipid that's being synthesized in a cell, right? That lipid that I mentioned is on the outside that we want to get rid of. And its role is to take that lipid and essentially use the power of ATP, adenosine triphosphate, to power a conformational change that now will flip it to the other side, okay? That's its role. It's a machine. All it does is take a molecule from one side and flip it to the other side. Sounds easy, right? Okay? And it's going to use ATP, and up here is the equation. ATP plus water goes to ADP plus inorganic phosphate, and you see a negative delta G. What does that tell you? It's spontaneous, right? That means there's energy in this molecule that I can consume that will power this change, right? This is how biology does it, right? In your cells right now, you are hydrolyzing ATP, right? And you have transporters that are doing similar processes, okay? So that's our goal. We want to understand this. Now it's becoming apparent that if we can inhibit or drug this particular transporter, we can kill gram-negative bacteria, right? So there's a lot of pharmaceutical companies that are trying to develop drugs that can bind to these different structures and essentially stop the machine from working, okay? So that's our goal. It's important for a drug target, and we really want to understand how the different structures impact, right, binding with drugs or lipids in the bilayer. So that's our goal. And what I want to do is, instead of showing you cartoons, I'm a biochemist. I love structure, right? I love seeing atoms and their positions. And I want to take you through what we know right now about how this machine actually works. So this machine is in the bilayer. So here's our bilayer. Here's these nucleotide binding domains. These are where ATP binds. And it now populates what we call an open inward state, where these are separated apart. And it's thought that this structure is open like this, right, to enable this cavity inside the transporter to have access now to that particularly bulky lipid, right? So now that bulky lipid can go inside this central cavity, and then the transporter will now come together, these nucleotide binding domains. I'm showing you the structure bound with an inhibitor developed at Genentech that locks this particular state. Now, at this particular conformation, I have ATP, and you've learned ATP has a negative delta G, and if I hydrolyze ATP, I can power a conformational change, right, in this structure. And if you focus on the top up here, you're going to see the change in conformation. So now I'm going to hydrolyze ATP, and now I'm going to power this conformational change that now enables that essential lipid or molecule to be transported to the other side of the bilayer, right? So in order to do this, I had to consume ATP, right? But I also have to bind that molecule. And so what I want to talk about today is how we're going to study this transporter, right, in biochemistry, and how we're going to understand and maybe reveal some new insights. All right, the approach, all right? We are biochemists that love mass spectrometry, right? I'll talk more about that. We love to weigh things, right? The periodic table, you saw masses. We like to determine the masses of proteins, right? We're interested in transporters, I'll talk about today. We're interested in lipids. We're interested in how ATP, for example, can impact these interactions and how it's important in the transport cycle. On campus, you should be excited about, we're doing a lot of this work, we're doing a lot of technology development, right, to push this field farther and ahead of our peers, right? And we also do a lot of traditional approaches, and I'll highlight some of that when we do structural biology, looking at structures at the atomic level, okay? All right, so just across the way at a building in that direction, my graduate students, your fellow classmates, right, Aggies, are doing this, all right? They're pumping out and performing all this work, this cutting edge research to generate discoveries, right, that help us understand how molecules work and how we can make better drugs, right? All right, mass spectrometry. So what we're going to do, as the Nobel Prize winner John Finn said, who developed electrospiralization, making elephants fly, all right? We're going to now take proteins, imagine those beautiful cartoons of those proteins I showed you, and we're going to fly them in a mass spectrometer, okay? It sounds easy, right? And so now we're going to introduce the proteins, we're going to fly them into a mass spectrometer, and our goal now is to measure their molecular mass, right? And we're going to try and measure that with really high precision, right? We want to measure it down to below a Dalton, right? And we can even measure it better depending on the size of the molecule. So today I'll talk about how we use this to measure molecular mass, and if a molecule binds our protein, what should happen to the mass? It should increase, right? And that's what we're going to be looking for, is looking at mass shifts, how molecules bind, right, in this mass spectrometer, okay? So using this expensive toy, research instrument, right, we can now understand how molecules work. My goal is to show you real data, because if I don't, you won't get excited about what's cutting edge and happening on campus. Here's mass to charge ratio, and here's the equation. This is essentially the mass of the complex where we've added protons to put a proton's a positive charge, right? And each of these peaks has a certain charge state. This is an 18 plus, and then the next one will be a 19 plus. All you need to know, it's very simple. If this is an 18 plus, I just need to multiply that MZ value by 18 to get the mass, right? Right? So now we have a way to measure the mass of this particular complex. Just to give you guys perspective, this took almost 2.5 years of work for one graduate student to achieve this, all right? This is actually quite a remarkable feat, because what Jixing, who's now at Regeneron, was able to show is that if you zoom in on this, right, and you look at the mass, this mass that we measure, molar mass, right, of roughly 129,000 grams per mole, right? I'll use Daltons, right? What we saw was 2 additional peaks, and these 2 peaks were offset by roughly 65 Daltons with a few Dalton mass error. And you, as taking first year chemistry, think about what 65 Daltons on the periodic table? Copper or zinc, right? And we could do a bunch of other experiments, and we've done that, and we were able to show that this is indeed copper. Copper 2 plus, by the way, binding this particular transporter. And if we add copper back, we could see binding of copper to the transporter, right? A very nice experiment. And I mentioned that this transporter is a dimer, and we see 2 binding of the metal. This indicates we have basically 1 binding per subunit, right? This is quite amazing. And just to give you perspective, this transporter I'm talking about has been studied for 3 decades, right? This is the first time that we've discovered it binds copper, all right? Pretty cool. All right, so now we need to understand 2 more things. We need to understand where's copper binding and how it influences binding with lipids, but we're also interested in drug binding. Here's a drug called G907. I showed you a structure earlier that was actually bound to this molecule. This is an inhibitor from Genentech to inhibit MSPA in that particular structure. And what we can show is that indeed we add the inhibitor and we can see binding of 2 molecules, 1 molecule per subunit, okay? We're not going to do much more on this inhibitor, but I'll show you how we're combining mass spectrometry to carry out a bunch of other biochemical experiments. All right. We're interested in copper, I mentioned, but we're also interested in affinity, right? And in chemistry, you're taught a chemical reaction, A plus B goes to C and D, right? I'm going to present that to you in a little different fashion. We're interested in how our protein, P, binds lipid to form a protein-lipid complex, right? And you, in the first year program, will tell me, that reminds me of that special ratio K. Remember, products over reactants, right? That special ratio of K at a given temperature is always the same no matter how I set up the system, right? And so we're interested in determining K because K is also related to delta G. And we'll come back to that. It tells us the strength of the interaction. And what we can do is we can take our mass spectrometer, we can add more of our ligand or lipid, and we can sort of dissect now what is the equilibrium binding constant, right? That special value of K that you've been learning about, right? And we can tease that out by generating a series of plots. All right, two things I want to point out. Here's a series of lipids, and I'm plotting K. Just think of a lower value means higher affinity. What I want to walk away on this slide is not all the gruesome details. What I want you to walk away with is how we can use this value of K to start understanding how the chemistry of the different lipids, how they impact binding to the transporter. So here I'm showing you values for binding the transporter with 0, 1, and 2 coppers, so partially metallated. And we can now do the same experiments for the transporter bound to metal, right? And the take home on this slide is that when we add metal, right, we now see a change in affinity for some, but not all of the lipids, right? This is quite surprising that metal, which hasn't been described for 3 decades, now can influence the affinity, right? So that's how we can use that special value of K to start understanding the importance of chemistry and how it impacts binding. So if we see that copper is influencing binding, what we need to understand is where does it bind, right? If it's doing something, how does it carry out its function? Okay? And so that's what we're going to do here. We've looked at these transporters in different structures, and we're trying to understand where is copper binding, right? And you can start thinking in your head, where do you think copper would bind? What residues in a protein? And so we've looked at this in many other structures. We don't see copper. And in fact, we see the N-terminal region of the protein in 1-letter format, as M is for methionine, H is for histidine, N is for asparagine, and so on. And this particular sequence, we surmise that this might be the region that's binding copper. And so what we can do, we can modify this on the DNA level. We can delete these residues and ask, does it bind copper? And when we delete it, it's gone. It no longer binds copper, right? Now here at A&M, what we really want to understand is, okay, if we can delete it, and it's important for binding copper, what does it look like, right? I want to visualize. I just don't want to know it binds this region. I want to know the 3-dimensional structure, right? And how do we do that? To give you an idea, I'm going to talk about what's called X-ray crystallography, right? And what we do is we grow protein crystals. Here's a crystal of a protein, right? And what we're going to do is going to subject them to high energy light X-rays. And our crystals that we grew here on campus, we shipped to Chicago. And by the way, this is the synchrotron. Just to give you perspective here, does everyone see these little cars? Yes? This is the ring. It's called the synchrotron. And in this ring, electrons are flying around at near the speed of light, right? Think about that. You can ride a bike. I've been there many times, right? But you can ride a bike around here. And we're going to use this synchrotron that's bringing these electrons around, and we're going to bend them in light, right, using magnets or a wiggler, right, to make them move in high energy light. And then we're going to expose that high energy light to our crystals to get a diffraction pattern. And from this process, we can determine the three-dimensional structure of our particular crystal, right? So that's what we did, and I'm going to show you how we've done that on a protein, a green fluorescent protein that now binds copper. We can now deduce the three-dimensional structure. This is what it looks like, okay? These are two views, 90-degree rotation about the plane of the projector. And what we can see is our three residues, methionine, histidine, and asparagine, right? And does everyone see the copper sitting right there in the middle? And you've learned about molecular geometry. You can tell me what kind of coordination that is, right? You see these beautiful planar interactions, these forms with the backbone of the peptide. And then you also can see two axial ligands, right? You have two waters, one below and one up top, that's being bridged by additional bonds, right? So now we have the structure to understand where copper is binding, right? And now we have the job to try and figure out how the heck is this thing modulating lipid binding, all right? But we at least know what the structure looks like, and we can continue moving forward. We can now do another thing. I mentioned all these beautiful snapshots or structures, right? We have the ability as biochemists to actually trap particular structures, and we can do this by playing with ATP. We can actually modify ATP in a way that it cannot be hydrolyzed, that it means it can bind the transporter, but we essentially now lock it in place. And you'll learn this in biochemistry as the transition state, or that big peak that you learned about EA in class. So we can trap this particular structure, and we can then now ask questions about how this structure now impacts or influences binding to other molecules, all right? And so that's what we've done here. We can do these experiments to now trap the transporter. We can then look at different values. Remember K, in this case, lower means higher affinity. We start to understand how trapping this particular structure enhances binding of some lipids, but not all of them, right? Now, some of you that have been paying attention in my lecture and in Gen Chem are probably saying, well, that's K at one temperature, right? It's not very useful because you didn't tell me what temperature you're collecting this data at, and you are at 37 degrees Celsius, right? Not room temperature, right? So one thing we can do is try to understand thermodynamics, right? And you've learned this, delta G equals delta H minus T delta S equals minus RT, and that natural log of that special ratio K, right? And we've learned that this is temperature dependent. And what I'm going to show you is how we can now back out using K, we can back out H and S. You've learned it in Gen Chem using two different K values at two different temperatures. We do experiments over a larger temperature range, right? And at some point you've learned about delta H, right? Bonds forming, breaking. In terms of what I'm showing you today, we're going to think about hydrogen bonds, electrostatic and hydrophobic interactions, and delta S as entropy, you've learned as randomness, right? Order to disorder. For example, when you first came here, your dorm room was probably perfectly ordered, right? What happened over time? It's chaos, I know, all right? That's entropy at work. And in terms of what I'm showing you today, we're interested in solvation, right? Where is our water molecules? Remember microstates, we're interested in dynamics or structures of proteins. Those come into play in entropy, right? And we're trying to understand how these terms tell us more about the chemistry or the molecular interaction, okay? Again, you're learning this in Gen Chem, and we do it here in actual real research, okay? All right, what we can do now, we need to control temperature. So here I'm showing you how we've developed a device where we have thermoelectric chips. We have our sample down here, and now we can control the solution temperature, meaning I can perform the measurements at different temperatures and record what happens, right? And you by now already know that every reaction that we study is temperature dependent, right? If I change temperature, I change the equilibrium, position, or that special measure K, and also talk about reaction rates. Here's some examples, right? So here I've got a protein on the left that is binding one lipid, and at low temperatures, 20 C, I see very little binding. If I take that same solution and I just heat it up all the way to 37, you can start to see more binding of that lipid, right? I'm not going to focus on the details, how many there are, but you will tell me that if this goes up, what's the driver for this? Entropy, right? So it's entropically favorable. We call this an entropy-driven reaction. On the right is the opposite. At lower temperatures, right, we see more binding, and as we heat up, we see less binding of the molecule, right? And you should know by now, instead of it being favorable, it's going to be unfavorable. Entropy will be negative, not positive, right? So reactions can be favorable in entropy and unfavorable, and that determines whether they proceed in the forward or the reverse direction, right? They also tell us how much we're going to have at an equilibrium position. And we can also pull out delta G, delta H, and minus T, delta S. And that now tells us what's the driver for the molecular interaction, whether it's enthalpy or entropy, all right? So again, the fundamental chemistry you're learning is being used in understanding biochemistry, right? All right, in the next remainder of my talk, I'm going to focus on nucleotide binding, right? I mentioned in the beginning that the transporter uses ATP to power conformational changes. You're probably wondering by now, how does ATP bind and how strong is the affinity? And another thing we want to understand is how does nucleotide binding influence binding to lipids, right? They're both coupled, and we want to understand this at a much higher level. All right, here's a mass spectrum again. I'm going to show you how we can now take this mass spectrum and start measuring in the presence of ATP. We can actually see binding of 1 and 2 ATP molecules. Remember that it had 2 nucleotide binding domains, right? And now we can see binding of 1 and 2 nucleotides. What we can do now is understand that special value of K, right, the equilibrium binding constant, and try to understand the affinity for binding ATP or ADP. And we can do that and pull out affinities, and we can show you that it binds with roughly close to 50 micromolar and 120 micromolar KD, right? We can also do these similar experiments to understand how ADP, not TP, right, diphosphate binds. The product of after ATP is hydrolyzed forms ADP, and we can see it binds with higher affinity, right? All right, so now I mentioned in the beginning that the transporter uses ATP to hydrolyze, right? But in this experiment, do you notice that there's no ATP being hydrolyzed, right? Essentially what we've done here is we've added ATP and we measure as quickly as we can so that no ATP is turned over. Doesn't this sound awfully familiar how you do kinetics? You set up a reaction, right? And you want to measure it as close to time 0 and start monitoring as close to 0 as the reaction proceeds, right? So that's what I'm going to show you in the next few slides, how we're going to now incorporate kinetics, right? Monitoring a reaction as a function of time, right? And I'm going to show you that here. We start off at time 0, so as close to 0 as we can. We add the nucleotide and we essentially now take the measurement. What we now want to understand is if we let this thing incubate, right? Doesn't matter how long, we let it incubate and turn over, what do you expect to happen, right? If ATP is hydrolyzed, what should the product be? ADP, right? And that's what we see. So if you look down at the bottom, you can see ATP binding of 1 and 2. And if you look down, we can see a peak emerging here that corresponds to 1 ADP and 1 ATP. By the way, this was really exciting to us because now it's telling us that the transporter, which has 2 nucleotide binding domains, is turning over 1 ATP at a time, right? Imagine an engine in your car. It doesn't fire all pistons at once. It fires them independently of each other, right? Same thing with the transporter. It's firing by using 1 ATP at a time, all right? Now, what can happen if we make this more complex? I talked a lot about lipids earlier, and how can we incorporate that into looking at kinetics? And we could do that. It's going to get a little bit more complex. I guess there's just more colors up here. So now we've got binding of lipid, and we see different ATP binding events. And this is now doing the same experiment. We now start at time 0, and we now let it turn over for 30 minutes, right? and ask the question, what's happened in the reaction, right? As the ATP is being hydrolyzed and turned over to power these conformational changes. I just want to highlight two really interesting things. And one thing is here. So here is that we're looking at a state that has 3 lipids binding of 1 and 2 ATPs. As the enzyme turns over, right, you can see that now you see it go up, and now it's binding more ATP. This is quite surprising. It's as if this machine's turning over and now adopting a higher affinity for its energy source, ATP. In addition, we can see over here the same thing when we have 4 lipids, right? It now also, when it turns over, adopts this high affinity state for the nucleotide, right? So this was quite surprising to us that we can monitor, one, the reaction in real time. We can watch the enzyme turn over, right? And we can capture all these states such as seeing ADP bound. We can continue to do this experiment longer than 30 minutes. We can do it over a longer time course, right? And I'm showing you different time points that we've collected measurements. And what we can see is differences in these peaks. That's all I want you to know, is that you see changes, right? Things are happening over time, right? And what's exciting about this is we can actually see this. We can actually watch the enzyme turnover. We can capture these different states, right? And try to understand, what does it mean in terms of the transport cycle, right? So far I've talked about thermodynamics, equilibrium binding constants, and kinetics. And I've talked about this transporter turning over, especially at this last part. What we want to do now is try and understand, can we get snapshots of this transporter in the transport cycle? Can we actually watch it turn over ATP in real time, right? That's our goal. So with these results, what we've then turned to is a technique called single particle cryogenic electron microscopy, right? This is a very expensive microscope, millions of dollars, right, that you're looking at here, right? That's actually across the train tracks right now, okay, on campus. And we're going to use this microscope, right, at cryogenic, just think really cold, okay, temperatures. And I'll talk about what single particle means in a second. So now we're going to take our sample, we're going to let it turn over nucleotide, and we're going to ask the question, if we flash freeze our sample, what can we see, right? And what structures will we find? And that's what we've done. So now what we've done is we flash freeze our sample after we let it turn over. And here I'm showing you what's called an electron micrograph, okay? A couple things. The magnification that we're looking at this is 81,000-fold magnification, right? In this micrograph, this is the scale bar, this is 90 nanometers, 90 times 10 to the minus 9 meters, right? Can you see little particles in here? If you look closely enough, there's actually these little transporters that I showed you at the beginning of my talk in this micrograph, okay? And this is one of almost 6,000. And what we have to do is go through this micrograph and pick out every single particle, right? If you look at that, you'll probably see what? A couple hundred, maybe 1,000, right? And we're going to go and pick out all these little particles out of these micrographs, right? And if we do that, you can imagine that we'll have millions of particles, and some may have different views, right? Some may be views of the side. And imagine if I had many views of the side, what can I do to those images? I could signal average, right, to get a better image, right? So the next goal will be taking all these different orientations of our protein in ice and their pictures and try and now average them. And that's what we're going to do here. Here's what we call 2D class averages. And if you look at this, do you see different structures, right? You can see some of these have the nucleotide binding domains open. You can see some side views. You can see some that are trapped here or in this closed state. Just to give you perspective, the box size of this square, each side is 273 angstroms, right? By the way, that's 10 to the minus 10 meters, right? That's the scale at which we're looking at these small proteins at the atomic level. Now, from all these micrographs, what we can do is take these micrographs and we can do some computation and generate a three-dimensional structure from these 2D images. And that's what we're going to do. What we're able to find is, here's 4 of these structures. I'm showing you the actual map. I'll just focus on the cartoon. We have 4 different structures that we can pull out of those different particles you saw on that micrograph. Okay. And what we see now is that these 2 nucleotide binding domains are coming closer and closer and closer together, right? We're essentially now catching the transporter in the act, right? It's part of the movement as it's doing its function, right? And we can also capture other structures. I'll show you one that I think is really exciting, and that's shown here. This is a structure that we've determined where you can see now it's got this open outward-facing state, right? And you can see the nucleotide binding domains on the bottom. By the way, guys, this is the real map which we as biochemists build an atomic model into, okay? So you can imagine taking this and putting atoms in here to generate your three-dimensional structure of your protein, right? And that's what we're doing. And we can generate a model from this. And what we found in this particular case is this lipid. I'm not going to get carried away, but just I want to make a quick point that this lipid we discovered eluded almost 3 decades of structural biology research, and that was discovered here on campus, right? And this turns out to be really important for function of this transporter. Not sure how everybody missed it, but at least we got it. And now what we can see is here we got the structure. We can now look at molecular details, look at more atoms, right? We can understand what is making this thing stick, if you will, to the protein, right? And you taking chemistry will tell me all about the molecular interactions. In this case, you can see electrostatic interactions, you can see hydrophobic interactions. These are the features that are important for recognizing this molecule and enhancing the binding to the transporter. There's some other interactions, but I won't spend a lot of time on this. Some residues in these transporters, we call them conserved, meaning in other sequences of other proteins, they're the same, and that's really important for function in order to recognize this molecule. All right. Let me wrap up with this and maybe a slide summarizing what I've showed you so far. What I want you to focus on is how we can understand how the transporter works. So now we've got this transporter in this open outward state. And if you look closely, hopefully you can see in red and orange is the ATP that's bound to the transporter, right? Now, this new structure I just showed you that we actually determined on campus now starts to tell us how the transporter releases nucleotide. So what I want you to do is focus on this bottom part, and I'm going to morph between these 2 structures. So I'm going to show you how we start with this closed and we go to this open state on the bottom, right? I'll rotate 90 for you, and I'll show you how we have it closed and it opens, and when it opens like this, it enables the nucleotide to be released from the transporter, right? This is important because after the transporter hydrolyzes ATP, what does it need to do? It needs to release, what's the product? ADP. It needs to release it, and it needs to reload with ATP, right? And so now we're starting to understand a more detailed picture of the transport cycle, right? So for time, I can't advance here. All right, so hopefully I've convinced you in roughly 44 minutes or so how we incorporate your first year chemistry and understanding biochemistry from special ratio of K, delta G, delta H, and delta S, and kinetics, right? And another thing I want to point out is structure, right? I showed you how we use different tools, X-ray crystallography and cryo-EM, to deduce the structure. And so what I've showed you today is essentially a view of this machine transporting this molecule, going from these open states to this closed state, to then binding ATP, to power a change into these structures, and then now populating a new structure where now the nucleotide is being released from the bottom, right, to now enable the transporter to now recycle and now do the process again, right? This is occurring in bacteria. It's also occurring in your cells. With that, I'll end by showing you a picture of the group, right? This is in ILSB and all the funding agencies and all the collaborators. All right, thank you.